Good morning, students. I am Professor Nitesh Sharma. Students, today I am teaching you differentiation during embryonic development. This topic, differentiation, is included in paper 3rd Developmental Biology of BSc Part 1st. Students, differentiation is the process by which one cell differs with other cell. So during cellular differentiation, the process of cellular differentiation is the process in which a cell changes from one cell type to another. Usually the cell changes to a more specialized type. Differentiation occurs numerous times during the development of a multicellular organism as it changes from a simple zygote to a complex system of tissues and cell types. So, students, cellular differentiation is the process in which a cell changes from one cell type to another. Usually, the cell changes to a more specialized type. Differentiation occurs numerous times during the development of a multicellular organism as it changes from a simple zygote to a complex system of tissues and cell types. Differentiation continues in adulthood as adult stem cells divide and create fully differentiated daughter cells during tissue repair and during normal cell turnover. So, students, some differentiation occurs in response to antigen exposure. Differentiation dramatically changes a cell's size, shape, membrane potential, metabolic activity, and responsiveness to signals. These changes are largely due to highly controlled modifications in gene expression and are the study of epigenetics. With a few exceptions, cellular differentiation almost never involves a change in the DNA sequence itself. Thus, different cells can have very different physical characteristics despite having the same genome. So, students, a specialized type of differentiation known as terminal differentiation is of importance in some tissues. For example, vertebrate nervous system, striated muscle, epidermis, and gut. During terminal differentiation, a precursor cell formerly capable of cell division permanently leaves the cell cycle, dismantles the cell cycle machinery, and often expresses a range of genes characteristic of the cell's final function. Example, myosin and actin for a muscle cell. Differentiation may continue to occur after terminal differentiation if the capacity and functions of the cell undergo further changes. So, students, among dividing cells, there are multiple levels of cell potency. The cell's ability to differentiate into other cell types. A greater potency indicates a larger number of cell types that can be derived. A cell that can differentiate into all cell types, including the placental tissue, is known as totipotent. In mammals, only the zygote and subsequent blastomeres are totipotent, while in plants, many differentiated cells can become totipotent with simple laboratory techniques. A cell that can be differentiated into all cell types of the adult organism is known as pluripotent. Such cells are called meristematic cells in higher plants, and embryonic stem cells in animals. Though some groups report the presence of adult pluripotent cells, virally induced expression of four transcription factors OCT4, SOX2, CMYC, and KLF4. Yamanka factors is sufficient to create pluripotent cells from adult fibroblasts. A multipotent cell is one that can differentiate into multiple different but closely related cell types. Oligopotent cells are more restricted than multipotent but can still differentiate into a few closely related cell types. Finally, unipotent cells can differentiate into only one cell type but are capable of self renewal. In cytopathology, the level of cellular differentiation is used as a measure of cancer progression. Grade is a marker of how differentiated a cell in a tumor is. So now, students, we are going to study the mechanism of differentiation. Each specialized cell type in an organism expresses a subset of all the genes that constitute the genome of the species. Each cell type is differentiated by its particular pattern of regulated gene expression. Cell differentiation is thus a transition of a cell from one cell type to another, and it involves a switch from one pattern of gene expression to another. Cellular differentiation during development can be understood as the result of a gene regulatory network. A regulatory gene and its cis regulatory modules are nodes in a gene regulatory network. They receive input and create output elsewhere in the network. The systems biology approach to developmental biology emphasizes the importance of investigating how developmental mechanisms interact to produce predictable patterns. However, an alternative view has been proposed recently based on stochastic gene expression. Cellular differentiation is the result of a Darwinian selective process occurring among cells. In this frame, protein and gene networks are the result of cellular processes and not their cause. So, students, while evolutionarily conserved molecular processes are involved in the cellular mechanisms underlying these switches, in animal species, these are very different from the well characterized gene regulatory mechanisms of bacteria and even from those of the animals closest unicellular relatives. Specifically, cell differentiation in animal is highly dependent on biomolecular condensates of regulatory proteins and enhancer DNA sequences. Student, cellular differentiation is often controlled by cell signaling. Many of the signal molecules that convey information from cell to cell during the control of cellular differentiation are called growth factors. Although the details of specific signal transduction pathways vary, these pathways often share the following general steps. A ligand produced by one cell binds to a receptor in the extracellular region of another cell, inducing a conformational change in the receptor. The shape of the cytoplasmic domain of the receptor changes and the receptor acquires enzymatic activity. The receptor then catalyzed reactions that phosphorylate other proteins, activating them. A cascade of phosphorylation reactions eventually activates a dormant transcription factor or cytoskeletal protein, thus contributing to the differentiation process in the target cell. Cells and tissues can vary in competence, their ability to respond to external signals. So, students, signal induction refers to cascades of signaling events during which a cell or tissue signals to another cell or tissue to influence its developmental fate. Yamamoto and Jeffrey investigated the role of the lens in eye formation in cave and surface dwelling fish. A striking example of induction through reciprocal transplants, Yamamoto and Jeffrey found that the lens vesicle of surface fish can induce other parts of the eye to develop in cave and surface dwelling fish, while the lens vesicle of the cave dwelling fish cannot. So, students, other important mechanisms fall under the category of asymmetric cell divisions, divisions that give rise to daughter cells with distinct developmental phases. Asymmetric cell divisions can occur because of asymmetrically expressed maternal cytoplasmic determinants or because of signaling. In the former mechanism, distinct daughter cells are created during cytokinesis because of an uneven distribution of regulatory molecules in the parent cell. The distinct cytoplasm that each daughter cell inherits results in a distinct pattern of differentiation for each daughter cell. A well studied example of pattern formation by asymmetric divisions is body axis patterning in Drosophila. RNA molecules are an important type of intracellular differentiation control signal. The molecular and genetic basis of asymmetric cell divisions has also been studied in Gree algae of the genus Volvox, a model system for studying how unicellular organisms can evolve into multicellular organisms. In Volvox carteri, the 16 cells in the anterior hemisphere of a 32 cell embryo divide asymmetrically, each producing one large and one small daughter cell. The size of the cell at the end of all cell divisions determines whether it becomes a specialized germ or somatic cells. So, students, now you are able to understand the process of differentiation. Thanks for listening and thanks for your patience.